Hi everyone, welcome to the JAX Tech Talk with the information scientist of the Jackson Laboratory. My name is Xintong, and my role as a technical information scientist is to serve the research community with educational and practical solutions. For anyone watching live, use the comment sections of the video to tell us where you are viewing from, and any related questions you have. We'll try to answer those live. So today, let's talk navigating the JAX Clinical Knowledgebase. And today I have my colleagues Sarah Edie and Song on the show with me. So first of all, I would like you guys to introduce yourself to our audience. What about Sarah, you go first?
Yeah, my name is Sarah Edie, and I've been at the Jackson Laboratory for just over 7 years now. And I used to be in TIS, so I used to be on the other side of the JAX Tech Talk, but I've recently moved to a new group, the JAX Clinical Knowledgebase. So I'm here to tell you about it.
Hello, everyone. Hello, everyone. My name is Som, and I'm a technical information scientist here at the Jackson Laboratory. My role at JAX is to assist researchers and research enthusiasts such as all of you who are listening in today by communicating all of your JAX-CKB queries with Sarah's team and understand how this can fit into your research goals.
Yeah, so Sarah, my first question, for our audience also, is to know what is CKB?
It's a great question to start. CKB is a relational database that can be used to interpret genomic profiles in cancer and identify potential treatments in the context of evidence and clinical trials. It includes content related to cancer, such as genes, variants identified in cancer, treatments, including targeted therapies and immunotherapies, and clinical trials. We make connections between the treatment, tumor type, and variants.
Well, it's good to know we have so many data curated within the CKB, and I wish I know this database when I pursue a PhD. That would be much helpful. So specifically, what kind of data is available and how much content that is accessible?
We have over 1,800 genes currently curated into the database that have been implicated in cancer. Each of these genes have curated variants with over 40,000 variants across all the genes. Additionally, we curate drugs that are either in clinical use or being developed preclinically for cancer therapeutic use. We curate clinical cancer trials from the US and Canada that are available on clinicaltrials.gov website and connect these trials to the genetic and indication requirements, allowing you to search by either drug, variant, or indication and find the relevant trials, whether they are actively recruiting and their locations. One big focus is what we call efficacy evidence. This is where we make the connection between a molecular profile, which consists of either single or multiple gene variants, a tumor type, and a therapy, which can be a single drug or multiple drugs used in combination, with a brief description of the data and response. Efficacy evidence allows you to look for therapies that may show a sensitive response in a particular tumor type in the context of a molecular profile or identify potential resistant variants.
Yeah, the most important question I do wish to know is where does this data come from?
We curate from published literature indexed in PubMed, but also from clinical trials, NCCN guidelines, and published abstracts. We have data ranging from FDA approvals, professional guidelines, to phase 1 through 3 trials, case reports, and even preclinical data. We include AMP, CAP, ASCO evidence levels for each efficacy evidence annotation, allowing you to sort or filter.
Nice. Yeah, that's really nice. So now I wish to take our audience to dive into the CKB to know more about it. And I'll have Sam to be our research scientist customer. So Sam, what questions you might have for Sarah if you are a first-time user of the CKB database?
Yeah, thanks, Xintong. So Sarah, imagine that I'm trying to find a list of BRAF variants for, say, NSCLC, and I wish to screen active and inactive variants from here to make a narrow selection list. What filters do I have to achieve this?
It's a great question. How about I show you by demonstrating? Give me a moment, and I'm going to share my screen. All right. Okay, so here is the JAX Clinical Knowledgebase. And in this, at the very front page, we have our basic search options. So for your question, I'm going to start by exploring by gene. So we curate our genes using the HGNC compliant gene symbol, but we do include all synonyms that are also typically associated with different genes. So as you start typing in the gene, you'll see that BRAF pops up here. And if I click on it and then hit submit, it's going to take me to the gene detail page. So here's our gene detail page. It's going to contain some information general information about the gene. So the gene symbol, the synonyms, and a 2-sentence gene description. The first sentence always describes the known function of the gene itself. And the second sentence will be relating that gene and its functions to cancer and how it's related to cancer. We also have some basic information underneath, such as the NCBI gene ID, chromosome map location, the canonical transcript we use, as well as the gene role, whether it's oncogene or tumor suppressor, and where we source that information. One thing I want to mention, as I mentioned previously, that our information does come from published PubMed indexed literature, and we link out to those papers that we use. So you see that all these papers, these PMIDs, are hyperlinked, and if you were to click on them, they will take you to PubMed, where you can find the abstract and source out to the paper itself. Now, for your question, Sam, you wanted to know more about, or if you want to find activating and inactivating mutations, variants that have inactivating and activating effects. So I think for you, the first column here under the gene detail, in the gene detail page, we have this gene variants tab. This gene variants tab includes the data about the gene variants that we've curated for this gene in our database. In the first column will be the variant, it's positioned within the protein. So at the protein level. The next column over will be the impact, so whether it's a missense protein, a fusion, a frame shift, for example. The next column over will be that protein effect, so whether we have a gain of function, loss of function, no effect. And so this is where you might find it to be really helpful. Now, for all of our data, we always provide places where you can sort or filter. So for example, you can use the arrows to just sort the data. But if you wanted to filter by a specific term, you can start filtering by typing into the search box up here, and it'll start filtering the data down to the variants that have a gain of function, for example. The next column over is that variant description. So this is where we're going to start having a 2-sentence description of the variant itself. The first sentence describes where the variant is located within the protein, and we use UniProt as our source for that information. And the second sentence will be the information that we've used to assign that we use for our decision to assign that protein effect. So what information we used and the data and the publications that we used to assign a gain of function effect. So you can use this section of the gene detail page to look for loss of function or gain of function mutants. Does that answer your question?
It does, Sarah. Thank you for that. And so I have another question. So let's say, can I use CKB to assist in my experimental design for finding information about molecules that probably already has demonstrated some sort of therapeutic effects? Let's say, for example, I want to understand how ATM impacts lung cancer. In order to select a positive control, can I use CKB and see a list of already tested therapies and hence use this knowledge to make the selection?
Yeah, so to do that, we can go back to the front page. So to go back to the front, if you ever need to go back, you can click on this little CKB icon at the top left. And we're going to go back to Explore by Gene. And I do apologize. Unfortunately, you're only seeing a portion of my screen. I'm not entirely sure why, but hopefully you'll still be able to follow through. I do apologize for this. As we know, in these days, there's always something that affects our ability to navigate technology. Let me try to stop sharing my screen and resharing. I just want to make sure you can see this because this is pretty important. So give me a moment and let me try this again. All right. Is that better? That's looking a little bit better. Okay, so we're going to go right back to what we did before, where we're going to search. You said ATM, correct? So again, as you see, as I start typing up the name, you're going to see the options pop down. Some cases like ATM, you may see there are multiple options because ATM is in the name of another gene symbol, but we're going to select the one that we know we're looking for and hit submit. Again, we're on the gene detail page. You can see it always starts on the gene variant tab, but I actually want you to look at this tab on the right, the gene level evidence tab. So I mentioned efficacy evidence, and this is where you're going to find the efficacy evidence for the ATM gene. Efficacy evidence, as I mentioned, connects a molecular profile to a tumor type, a therapy, and a response. So starting from the left, you're going to see the molecular profile. This can be one or more gene variants. In some cases, we have used what we call category variants. Category variants allow us to stay true to the literature. So sometimes in guidelines or papers or FDA approval, we don't have a specific variant that's been described, but it may be describing a group of variants. So for example, this molecular profile is for ATM inactivating mutations. So in order to be able to address that, since it's not a specific variant, we created these category variants to account for that information and be able to curate this information in our database. So we have our molecular profile. The next column over is our indication or tumor type, which we source from disease ontology. We have a response type, so it can be resistant, sensitive, no benefit, not applicable, for example. Those are some of the ones you might find. The next tab over is the therapy. Again, I mentioned the therapy can be either a single drug or can be multiple drugs used in combination, as you see here. And we have the approval status. So this is, we source data, as I mentioned, from multiple different locations. So we have FDA approvals, we have guidelines, phase 1 through 3. And if you were to scroll all the way down to the bottom, you're going to see preclinical data as well. And just like before, you can always sort through any of these columns. And in this tab here, where it says efficacy evidence, this is the summary data that we use to assign a response type to the molecular profile, indication, and therapy. So this will be a summary of a paper, abstract, or guidelines. And again, we always link out to where we source our data from. Now, you said you were looking for a positive control. So you're looking for something that may have a sensitive response. So as we start typing, it'll start filtering the column. And as you can see, we have some therapies that show a sensitive response with one or more ATM molecular profiles and for different tumors. Now, you specifically mentioned lung, so we can look down into looking more closely at what lung tumor types we might have. As you can see, our approval statuses have changed, so we don't have the FDA or guideline level. We have case reports and preclinical data. Now, this may be something that you can now use to select a positive control, or if you want something that has a little bit higher approval status, you might try removing the lung filter and look at all the data that comes from other tumor types as well. Does that answer your question, Sam?
Yes, it does, Sarah. This is really, really powerful and a very nice demo, very helpful. Last question, how can our viewers access the CKB interface?
Again, a great question. So I've been using the CKB core. We do have a couple different options. So we have a link, first of all, to the database in the description box. So whether you're watching from LinkedIn or from YouTube, you can see it in the description box, a link to CKB Home. So CKB Core, which is what I've been using today, is free for customers. It provides access to the content for 50 genes with more than 8,000 variants curated to those genes. For access to over 1,800 genes and 40,000 variants, we have CKB Boost, which is a paid subscription to the complete web-based content, along with some advanced search options and visuals. Lastly, if you are interested in integrating the data into a pipeline, we have CKB Flex, in which we send the database as a JSON or flat file on a weekly basis. If you have any questions about these options, or if you want a more in-depth demo, please contact CKB support at jax.org.
Yeah, Sarah, nice. If you could reshare this page, looks like we only see part of the page of how to access to that.
Oh, unfortunately, it's one of those situations. I feel like whenever you're sharing a screen, something.
And again, I want to thank Sarah and thank Sam. This conversation is really nice, and this nice talk definitely is informative and helpful to understand how to use CKB for preclinical and clinical study applications. And most importantly, as I am a cancer researcher and I do know how valuable this data could be useful. Like I said, the curated genes may be useful as a biomarker for cancer detection, and those proved efficacy studies may be used as a rationale to design a bispecific or different antibody-based immunotherapies. So that's all the time we have for this week. And I do share 3 links in the LinkedIn page and the YouTube. And you'll find that the first link, CKB homepage, will lead you to the CKB database and let you know how to get access to the database. And we also have a resources page and the clinical knowledge CKB page. These 2 pages share with you about the general introduction of CKB, as well as what the data are readily available from this page. So I want to thank you for joining us this week, and our next JAX Tech Talk will be scheduled on January 22nd, 2022. And the proposed topic is about NSG double knockout mice and its applications for graft versus host disease. Please follow JAX Tech Talk on LinkedIn so you are notified anytime we have a new episode, or subscribe to our playlist on YouTube. And this is Xintong saying stay healthy, stay safe, and stay excited about research. Bye-bye, everyone.
Bye.
Bye.